I am thrilled to welcome you all to our Allan Institute for Brain Science 2010 Annual Symposium, Open Questions in Neuroscience. I'll tell you a little bit more about that after I tell you a little bit about the Allan Institute in my talk. But again, welcome everyone. So let me just kick off by giving a little bit of an intro to the Allan Institute for those of you who don't know us and what we do. I'll work out some of the kinks here, and I'm supposed to, one of the key rules today is for me to keep the timer going so we can keep on track with our time, and I'll do that to myself as well. So if I go to, is this going to advance? Nope, that didn't do it. Is there a wireless mouse?
It's the advance button on the right.
Okay. There we go. All right. So the Allan Institute, this is my slide I always give at my talks, and it's who we are and what we are. So when I'm in Seattle, it's a little bit easier because I think many of you know who we are. We are an independent, we're not-for-profit research organization, and our core mission is really to support basic research around the world. We do this by making tools, data, information readily available to scientists around the world, and we do that all for free. The Institute is structured much more like a biotech organization than a traditional research organization that you might find. So we're very much milestone-driven and project-focused. And I'll tell you more about the large-scale projects that we've undertaken and will be undertaking in the future. So to do all of this, we have this unique ability to assemble a whole bunch of different great talent across many different disciplines under one roof, all geared towards delivering these products for the scientific community. So we like to talk about us being both multidisciplinary here, working towards these common goals. We have PhDs in math, physics, engineering, systems level, and molecular neuroscience. And again, all of these guys are working together to create these resources. A question that I commonly get asked, how many people are at the Institute? As of last Friday, I believe, we have 140 staff. Actually, one more person joined today and is actually attending today. So we now have 30 PhDs on staff. We are located in the Fremont neighborhood of Seattle, so right in the middle, right on the canal there. Have a nice space that's mixed lab and office space. Okay? And what we are not is a traditional PI-driven organization, so we're not problem-focused per se, and we're not an extramural funding agency. So the Institute was founded back in 2000, 2001, and the original planning meetings were really focused, I think, on these very big problems in neuroscience. How does the brain work? And really thinking about consciousness and thought. As we often do, and as we're doing today, Paul convened a group of experts, the world's experts, and said, where should we start trying to tackle this problem? And those experts, some of whom are in the room today, really recommended starting at some fundamental and moving forward across here. So one can imagine that our overarching goal, and I'll show you some of the projects we're doing, and you can see this kind of movement, is really to start here at these fundamental building blocks and move very systematically, do this in industrial scale fashion, very systematically build up knowledge around how genes function in the brain, then moving to concepts of understanding cell types, then starting to think about connections, synapses, computation, ultimately getting up to things like consciousness. But this is a very, very long-term goal. And as you'll see, we have projects now that have sort of tackled this reasonably well, just starting to do this and contemplating this. Okay? So our platform that we've created at the Allan Institute is largely tissue processing. We process brain tissue from a variety of different species, also things like human glioblastoma. And it's largely a high-throughput tissue processing facility where we can do a number of different techniques in very high-throughput ways using industrialized tools, things that were mostly off the shelf. We've done some custom automation of our own. but basically in a process where we can generate over 4,000 slides in a given week. And a key part of this is not just generating the wet lab slides, a microscope slide as you see here, but actually turning it into something in silico so that people around the world can come access that data, mine it, and use it. So once we've created that in silico data, we of course need to process it. There's a whole team of informatics and as well as information technologists who are helping us create these pipelines for managing all of this data, for processing it, and then ultimately we serve it out to the public for free on these web-based viewers. Okay, and this is the general overview of the Institute and our technologies. Now, these movies are not going to play, and I did test them before, and they're going to disappear. But I'll just see if it's even going to advance that. Okay, unfortunately, the movies are not playing today. I guess this is a good thing why people brought their own laptops. So the Institute itself, the Institute website is brain-map.org, and I'll show you the different offerings that we have here. But effectively, there's about 1,000 unique users that come and access that data every day, and that sort of just demonstrates how useful this data is to scientists around the world. We don't actually track, we don't require people to register to use the data, but we are able to track URLs and that sort of thing. And so every major pharma company uses this data, and really it's around the world usage of this data. And I would show you some of the highlights here in the tools, but you should go check them out yourself. Brain-map.org. And let me see if I can get this to even advance. There we go. So this just highlights over time. Here we sit today in 2010, and we've already committed to additional scope for various projects. I'm going to walk through each of these very, very quickly to just kind of highlight the range of activities that we've got going on and give a little food for thought about where we might be going and talking about this symposium. So we started out with the Adult Mouse Brain Atlas. This was a gene expression atlas using in situ hybridization. We added a number of other projects, and I won't talk about many of these, but we're doing large-scale efforts we've done on the mouse spinal cord effort, developing mouse brain. We've begun human work in 2006, working first on the neocortex, and then doing the whole adult human brain, and I'll tell you more about that project later. We're doing work on non-human primate, glioblastoma, and then recently NIMH-funded work on the developing human brain. We're also launching new projects this year on a mouse connectivity atlas and one on a networks of information within cells, and this is just in the very early planning stages. Okay. So to briefly go over the large-scale institute projects, this highlights the mouse brain atlas. So we had created using in situ hybridization, going systematically through gene by gene, looking at gene expression in the mouse brain, and then map that back into a 3D coordinate system so that we get this 3D map at the end where all of the genes have been mapped into that framework. And that allows us to do interesting things like mine the data now. So we've actually we've generated all this two-dimensional data, but mapped it back into the 3D framework. So now we have something on the order of 50,000 voxels or something in volumetric space across 20,000 genes. And this is a huge data matrix, which allows you to do all kinds of correlations and other studies to start to link anatomy with the underlying biochemistry of the brain. Go to the next slide. This is the Allan Spinal Cord Atlas. So when we were completing the Adult Mouse Brain Atlas, one of the researchers in spinal cord community said, why aren't you doing spinal cord? You should put it on the slides. We would really value having this information. And that ended up being a sort of a community-wide effort. There were actually a number of different funders who funded that particular project. And in very quick order, because we had all the machinery built up to do this, we were able to go through the mouse spinal cord, both in juvenile and adult, looking at about 20,000 genes, I guess. Okay. Okay. Developing Mouse Brain Atlas. So this is a project that we began about 2.5 years ago, completed earlier this year. and we looked at about 2,000 genes across 7 different stages of development. So this was, these are genes that are really heavily involved in development, transcription factors, other things that people were particularly interested in seeing. And we also created a series of reference atlases to map this into. And we're currently now proceeding with the mining of all of this data. It's very, very rich in terms of understanding the transcriptional code for development. Because we ran all these transcription factors through, and now we're doing automated mapping back into our reference framework and being able to do interesting mining activities with that. Another project that we've done that really isn't atlases, but I think has interesting and different impact on the community, is the creation of a number of different transgenic reporter lines, and this is done by Honghui Zhang and her R&D group. But effectively what we're doing now, and this list extends now to AI, something like 32 or 38 or something like that. It's a whole series of different toolkit for people doing transgenic mice experiments. So we've been building out these lines over the last several years, and they become very popular. And I'm just going to highlight, this is, again, a different way. You imagine 1,000 unique users every day using the mouse brain atlas data and mining it. However, you can also imagine the utility, and many of you are researchers in your own labs, use transgenic mice or order them, et cetera. So I just like to highlight the timeline where Honghui and Ed Lean both had been building up these lines over time. We actually provided breeding pairs to JAX so that they could start to be distributed well before Honghui ever published a paper on that in January of 2010, such that as of just last Friday, 1,500 of these reporter mice had been distributed either by JAX or by us to researchers around the world. And so I think it's a good model. We didn't get scooped, and it really shows the kind of impact and the demand that creating good tools for researchers can have. Okay. So we're going to build on these Cree reporters, or these Cree driver lines that we've created. So we imagine that we mapped all of the adult mouse brain information that told us about specific genes that were focusing activity within specific cell types in the brain. That gave us a genetic handle to go build these drivers to be able to make Cree drivers and then put reporters into them. Those reporters can be things like channel rhodopsins, halorhodopsins, fluorescent reporters, things that will actually kill the cells, and that gives you a real good genetic handle on that. The other thing that we can do is take advantage of that by using viral tracers, which are Cree dependent, and we're actually going to do a very large-scale project now where we're mapping the wiring of the mouse brain, and we're doing that by leveraging all of those Cree lines and then going in and very systematically going in various different parts of the brain and building up this data set over time. This is a project that we're just launching now, so we're in the early data generation stages of this, but we'll be ramping up soon to do massive amounts of this. So we've also then switched to do both human and non-human primates. The non-human primate work has been funded by the NIH and has been done in collaboration with various groups. And we're also doing the developing brain. And these projects, this is the non-human primate work. This is basically in situ hybridization across early postnatal development, looking at various changes in structures on a small number of key genes of interest. The Transcriptional Atlas of Human Brain Development, this was a large project funded by the NIMH as part of the GO stimulus grant money. This was a project that was a joint effort between Yale University, USC, and the Allan Institute. Yale and USC are doing a lot of transcriptional profiling, deep sequencing, and then we're creating atlases and other structure. We're working with Bruce Fischel at Harvard to do MR atlases and combining all of this stuff and presenting it here. So this resource is going to have its first data release in October. This is a very short duration project, but we're well on our way to creating yet another useful resource. we go. So a few fun facts. A lot of times when people come onto the website and they maybe access a few pieces of data, they don't really get a sense of the scale of what we're doing. So I created this slide to just give a sense of the scale. Since inception, we've generated data on over 2.8 million tissue sections. There might even be some of the people in here who have actually painstakingly done that work. That's one of the areas that we haven't able to actually automate. So each one of those 2.8 million tissue sections has been put on by hand by like one of the technicians here who are fantastic. We've processed over 625,000 microscope slides over the course of our projects. So this would stretch from Fremont to Tacoma, so it's quite a lot of microscope slides. We're over a petabyte of raw image data at this point, which is a lot of data storage if it was put on magnetic tape. One of the things that we got early on, and this is less of an issue as the internet has gotten more robust about serving large data sets, but there's a mirror site that's part of this International Informatics Consortium that's in Sweden to create a mirror site. And then we're also looking at creating a mirror site in Okinawa to serve up Asia. We've also created what's called an API, which allows people to come in and grab the data at the sort of the guts level. So for people who know what they're doing, who don't want to use the web tools, but rather want to get into the guts of the data and pull it and use it in unique ways, we've enabled that so that people can create these mashups, and that's resulted in all kinds of fun things, including things like translation of our pages into various languages. And finally, just highlight this bullet here. In May of 2010, we released our first data set on the adult human brain. This is 48.5 million gene expression data points on a single adult human brain. All right. So this highlights what we've done for the Human Brain Atlas. Effectively, we're starting out with, these are cadaver brains. They are, but they have to be fresh. So there's a lot of timing issues for getting a normal human brain. We do MR and DTI to create a framework for that, and then we start partitioning the brain. It's first slabbed and then frozen, sent to the institute, and we do a series of histology to get the anatomy just right. Then it's further partitioned, and then based on the anatomy, we either do laser capture microdissection to isolate subcortical structures, or in the case of the cortex, we do a macrodissection with a scalpel. Ultimately, we're creating about 800 to 1,000 samples per adult human brain that we're then putting on a microarray to get a gene expression readout. Okay, so we start out with whole brain, partition into cerebellum and brainstem, and then do the further partitions based on the anatomy. This shows a macrodissection workflow where we're collecting these regions. And then ultimately, this goes on a microarray. Painfully, we may need to bring the laptop down here because it's not advancing. There it goes. To get these sort of expression patterns. And if I just go through here, this is what an LCM workflow would be for something like the hippocampus. Okay, so the regions are highlighted here and then collected here. And we provide all of this information, this sort of metadata, when we serve up the web tools. Okay? And so the web tools, we did a first release in May. We've got another release in October that's going to have start at least some samples from the second human brain that we're processing through. It takes us about 6 to 8 months to process a whole brain from start to finish. But there's a lot of nice tools for being able to look at any kind of different associated data that people like to look at. Histology, MR with anatomic assignments. We'll be releasing a 3D viewer where you can actually look at the gene expression within a 3D context, where we've actually taken the samples, et cetera. And what this allows people to do, I think for the very first time, and it's really pretty remarkable, is the ability to go in and look at something like ion channels. Okay, so this is 100 ion channels across basically 500 samples across the entire brain. And this is just highlighting the unique characters of all of these things, but such important molecules for the functioning of the brain involved in governing electrical activity of the brain, you can see the diversity across the brain. You can see areas of clustering. All the anatomic regions cluster very nicely. And I think it just shows the power of these kinds of resources for people who are interested in any kind of different biology related to the brain. Okay. So just a very quick look at what people have started to do with this. So one of the things that we were excited about is being able to link genes to function. And Michael Grecius at Stanford University has been collaborating with us to take a look at this. He's been looking at basically functional connectivity networks in resting state. Not to go into too much of the detail, but intriguingly, it looks like there will be gene clusters which seem to cluster with areas of activity in the sort of default mode network. So very early analysis, but it looks very promising that we might be able to make some links. We're also then beginning our phase 2 work, which is actually to go in and do our in situ hybridization on the human brain, and more of that to come. Our final product is going to have a combination of microarray data, comprehensive, as well as in situ hybridization. Okay, so now I'd like to spend the next few minutes just setting the stage for our symposium for the next day and a half, and just briefly talk about, so I just went through the large-scale projects, and they tend to be the lion's share of our activity at the Institute. But we also have this sort of productive pipeline here where we're doing a number of things in sort of mining that data, trying to publish our own papers on it, but provide unique demonstrations. The kinds of things like creating these transgenic mice came out of those efforts of mining the large-scale projects. We call those trailing edge activities, so they're sort of after we've done the big project. But those feed back very nicely, so something like the transgenic animals feed back to something that we then put into piloting stages for something like the connectional atlas. So we did piloting, proof of concept, to evaluate whether or not there was something there that was worthwhile scaling. And indeed, it looked like there was, and so we're now moving forward with the large-scale projects. So one of the things we'd like to get out of the Allan Institute Symposium are additional ideas. What are those things that really, Paul likes to say, move the needle, things that can really influence and move the field, that are things that are pretty unique for the Allan Institute to do. So these are not things that can be done elsewhere, but really sort of take advantage of the unique culture and resource that we've created. And so as part of this is you sort of to capture a lot of ideas, food for thought to think about either technologies or proof of concept or other things that might be valuable to consider in moving forward. So with that, we will advance, hopefully, to the final slide. So, of course, none of the work that I talked about was done by me. It was done by a great group of people, in addition to a huge number of other science advisors and other things that all contribute in various ways collaboratively to our efforts. But key among them, and this is something that every time I give a talk, I thank Paul Allan. He's actually in the room today, so it's a rare opportunity for me to actually thank him publicly. We should all give him a round of applause, I think, for... It's really to make these transformational investments and to continue to do them, I think, is a rare testament to what Paul's been able to do. We're thrilled to be able to deliver that, and we're thrilled to have you guys here to help us in thinking about what might be done in the future.